Oi, me escutando? Oi, Clarice, eu vou só algumas mensagens aqui para o colóquio, para o pessoal do auditório. Então, a gente vai começar mais um colóquio do IFSC hoje. Só que antes de começar, a gente vai pedir um minuto de silêncio pelo falecimento de um colega nosso, o funcionário Paulo Villani, que faleceu ontem. Então, a gente vai fazer só um minutinho de silêncio em homenagem ao Paulo, que era um colega nosso, muito querido aqui no Instituto. Então, só queria pedir um minutinho de silêncio e depois a gente dá início ao colóquio, tá bom? Obrigado a todos. Bom, então, com isso, acho que podemos finalmente dar início ao nosso colóquio. E hoje nós temos o prazer de receber a professora Clarice Aiello. Eu vou fazer uma pequena introdução da Clarice. A Clarice é brasileira, está há muito tempo fora. Então, a Clarice ela tem bacharelado em Física pela École Polytechnique, na França, tem mestrado pela Universidade de Cambridge, no Trinity College, e doutorado em Engenharia Elétrica pelo MIT. E ela também realizou pós-doutorados em Bioengenharia na Universidade de Stanford e Química na Universidade de Berkeley. E desde 2020, 2019, 2 meses antes da pandemia, ela ingressou na UCLA como assistant professor, e hoje lidera o Laboratório de Tecnologia em Biologia Quântica, o QBIT. A Clarice ela é engenheira quântica, interessada em como a física quântica informa a biologia em nanoescala. Ela é especialista em nanosensores, que aproveitam os efeitos quânticos da temperatura ambiente em ambientes ruidosos. E hoje a Clarice vai falar para a gente um pouco da quantum biology, how nature harnesses quantum processes to function optimally, and how might we control such quantum processes to therapeutic advantages. Então, obrigado, Clarice. Agora o palco é teu, você pode começar. Obrigado.
Gente, obrigada por estar ouvindo, por estar aí ouvindo a minha palestra. Eu vou encorajar vocês por favor, me pausem durante a minha palestra para fazer perguntas, assim a gente fica mais um diálogo, né? Eu vou fazer a minha palestra em inglês, porque o meu português está meio broke. Tá meio ruim. Eu não sei se eu sei falar ciência em português mais, mas, por favor, eu vou responder pergunta em português. Fiquem à vontade para, de novo, fazer pergunta em português. A gente faz uma mistura meio macarrônica aqui de inglês e português. Vocês estão vendo a minha tela? Tá, eu vou assumir que tá.
Sim, então.
Então, tá. Ok, então vamos lá, gente. Eu tô aqui para falar para vocês sobre sobre a biologia quântica. So bear with me a little bit. Let's see where I'm going with this. So I like to call myself a quantum engineer. So this means that I build instruments to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us. So people get a little bit confused when I say that I work in things that might be happening inside birds, inside butterflies, inside our own bodies. So before I explain where I'm going, I really need to tell you where I come from. We're going to start with straight quantum mechanics, but by the end of the hour, we will have talked about things of biological relevance, such as organismal migration, how our cells respond to stress, to oxidative stress, to radiation, and I hope I will have convinced that I think we can learn with nature how to build better technologies. And that's because I'm a quantum engineer interested in how quantum physics informs biology at the nanoscale. So I always like to start by saying that I think humankind is obsessed with measuring things better because this means that we might understand nature better in general. For example, we might want to measure better frequencies or to have a better definition of what the second is. This machine that you see there is an atomic clock that sits in an American institution called NIST, the National Institutes of Technologies and Standards, and it uses very precise atomic transitions to define what a second is. We can think about measuring better magnetic fields so that the image of your baby is better resolved. And yes, that's the magnetic resonance image of a baby inside the mother's belly. Or we can think about using measuring better accelerations with those tiny little accelerometers so that our gaming experience is enhanced. So those tiny little accelerometers are now ubiquitous in all our handheld devices. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity that you want to measure is very, very small? I'm going to argue that in this case, you need a tiny little sensor that can measure tiny little things. If the sensor is tiny, let's make it very tiny. Let's make it quantum. For reasons that I won't have time to explain, you can actually prove that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances your measurement, the result of your measurement. I'm going to spend the first part of my talk talking about a technological quantum sensor that I worked with in the past. So the reason why I talk about this technological quantum sensor is mostly to draw comparisons to another type of quantum sensor that I'm going to claim is nature made. So I'm going to be drawing a lot of comparisons between these technological quantum sensors and the quantum sensors found in nature that I'm mostly interested in right now. But in this first part of the talk, the premise is that one can put a single electron spin in diamond, in the material diamond that some of you might have in your jewelry, to work as a quantum enhanced magnetic field sensor. So let's see how that works. The particular electron spinning diamond that I worked with arises, it's sort of an effective electron that is strapped into a crystal defect of diamond. Diamond has many different crystal defects. The defect that I worked with is called a color center. Color center are the crystalline defects responsible for the colors of those very nice diamonds that you see there at the bottom of the slide. And those defects are called color centers because when light impinges onto them, they absorb this light, get excited, and then de-excite by emitting fluorescence. The particular defect that I worked with is called a nitrogen vacancy center. It arises naturally, but it can also be engineered, and it happens when a vacancy, a missing carbon atom, sits nearby to a nitrogen atom, which is the most commonly found substitutional impurity in the carbon lattice. When those two things come together, the substitutional nitrogen and the missing carbon atom, so there's a lot of weird things happening, right? So first of all, there's like unpaired electrons close to the vacancy. There's an extra electron given by the nitrogen. There might be charges nearby coming in and out. But something very convenient happens when you start calculating the quantum mechanical energy levels of this electronic compound. And it turns out that the quantum mechanic energy levels of this electronic compound correspond to the energy levels that we would find in a single electron spin. Actually, in fact, it's an electron spin 1, not electron spin 1/2, as we're used to, but it doesn't matter. We can think of this atomic mass as a single effective trapped electron spin that is trapped locally there and doesn't move. So that's how we actually think of this defect in diamond. And the way that we play with this defect in diamond is that we have a diamond sample. Everything is at room temperature, no vacuum required. We have a diamond sample. The very first diamond sample that we worked with was a discard from a jeweler. So nothing particularly of importance there. Now people are making those materials much better and all, but in principle, this occurs naturally. Diamond sample on top of a moving stage, and then you move it around at the same time that you excite this diamond with a laser of an appropriate color. So as you're scanning, if the laser hits one of those defects, it's a color center, so it absorbs light, gets excited, and then de-excites by emitting fluorescence. This fluorescence we can collect with single photodetectors, and this fluorescence, this is what you get for this fluorescent signal at increasing length scales. So in the bottom right picture, you see here what's effectively the fluorescence signature of a single effective electronic spin. So the defect is effectively atomically sized, but this is a diffraction limited image, so that you see a little blob of about like half a micron or something. But this is very unusual because, I mean, if you're familiar with magnetic resonance methods, for example, you need a gazillion spins to get an appreciable signal. And here you are actually capturing the fluorescence signature of a single electron spin. So that's quite remarkable already. But it actually gets more remarkable than that, because this particular defect in diamond, this particular trapped electron, has a property that is called, that in jargon, people in quantum physics call a quantum state dependent fluorescence intensity. This is what this means. So this means that just by looking at how strongly this blob is in your computer screen, how much light is being emitted, you can actually infer what the electron spin state of your diamond is. So, as I said, this is a effective spin 1 particle, so that it has 3 magnetic sublevels, -1, 0, and +1. And what people do is they choose 2 of those out of those 3 levels and make an effective 2-level system out of it, right? And it can be shown that if the spin is in this state, and now you can think of it as an effective quantum bit, an effective spin. If spin is up, you have a certain fluorescence intensity. If spin is down, you have another certain fluorescence intensity. So that just by looking at how strongly this is fluorescing, you can actually do what's called quantum state readout, which is very convenient. It's determining the quantum state via a fluorescence intensity, via light intensity. And we can make very good photon detectors to help us with this task. But I said we could put it to use as a very sensitive magnetometer. Here's how this works. Again, those are 2 of the magnetic sublevels that I was talking to you about, that we make an effective 2-level system out of. Level number 0 here, magnetic sublevel 0. Oh, sorry. Their resonance frequency is in the microwave regime. It's about 3 gigahertz. But magnetic sublevel 0, it shouldn't interact with magnetic fields. So if you place your diamond into a magnetic field, magnetic sublevel 0 will not care. Whereas if the spin is in magnetic sublevel +1, as you see here, in the presence of a magnetic field, due to the Zeeman interaction, this magnetic sublevel +1 will be shifted, will be perturbed by this magnetic field by a quantity that I'm calling delta here. right? So, and it turns out again, because of the Zeeman interaction, delta is proportional to the magnetic field strength. So if you're sleeping, now is the time to wake up, because what I'm going to tell you now is like 2 minutes of how quantum sensing works. So here's what's interesting. Now, and I want to make sure to make this point because I don't think it's sufficiently made. Measuring magnetic field is effectively mapped into the problem of the experimentalist being able to measure a detuning, a shift from a known resonance, the energy difference between 0 and 1, as a function of magnetic field, or as a function that you want to measure. This actually relies, say, 80% of quantum sensing modalities. There's a certain quantity that you want to measure, magnetic field, temperature, electric field, that shifts one of your resonances. And if the experimentalist knows how to measure this energy difference, you can actually have access to the quantity itself, right? So this is a crash course in quantum sensing. So I like to think of it as a, if you will, an electric a signal processing problem, because if you know how to measure your detuning, your shifting resonance from your signal, you have access to this very small quantity that you want to mention. For reasons that I will not have time to explain, but it's cool. Experimentalists have known for decades how to measure such detunings, and it turns out that measuring such detunings from resonance involves putting the spin into a superposition state of 0 and 1, and seeing how the relative phase between those two states evolve. So here's the quantum part of it. This relies on the spin being put into a superposition state. So, but again, this modality, because it involves superposition, it only works while the spin is very well described by the properties of the laws of quantum mechanics. Everything that starts quantum dies classical. And remarkably, for this effective spinning diamond, the typical decoherence times for the experts in the room, I'm quoting T2 star for naturally occurring diamond, is about 2 microseconds at room temperature in bulk. And if you work in quantum and quantum information, you will know that this is extremely long for a quantum object to remain, for the spin to remain well described by the laws of quantum mechanics for 2 microseconds. That's extremely long, and that's one of the reasons why people like this sensor so much. So remember this number, 2 microseconds. So again, as I was saying, I like to think of quantum sensing as a signal processing problem, right? So you have a signal as a function of time, and the idea is that you have a certain signal, and the moment that your sensor dies as a quantum object, the signal ceases to oscillate or to do its thing. It flattens out. So it doesn't matter how much more you acquire your signal for, because if you do the Fourier transform of the signal, you don't get a better resolution in the spectrum. So a lot of what physicists spend their time thinking about is how to extend the quantumness of this spin so that you have the signal going for longer and longer. And a lot of you might know that you do this with engineered electromagnetic fields, right? There's like light matter interaction, so that if you can give electromagnetic kicks to this spin, you can keep it going longer as a quantum object. If you're just starting and you haven't seen this yet, think of it as a swing in the playground. If you give it kicks at particular times, you can keep the swing going back and forth. We can do the same for a quantum system. So with light, with engineered electromagnetic fields. I'm going to show you one instance of such kicks, electromagnetic engineered kicks. I'm going to go for possibly the most technical slide in my talk, so don't lose overview of the big picture here. The big picture is that we have a tiny little sensor that we bring in close proximity to a tiny little sample, and we're able to read out results from the tiny little sample, magnetic fields produced by this tiny little sample. So this is the big picture. That's the picture that I want you to remember. So here's one example of such a spin control experiment. So in particular, the electromagnetic kicks that I applied onto my spin so that it lived longer as a quantum object is called a rotary echo sequence. So the rotary echo is depicted in nuclear magnetic resonance jargon to the right. Here's what this means. It means that I have my diamond slab and I bring a microwave cable with a microwave signal that is tuned to your original two-level system frequency, about 3 gigahertz. Okay, the microwave is always on at that original original frequency, but at particular time intervals, you sort of give phase kicks to the electromagnetic radiation that you are applying. So this constitutes a pulse sequence to control your spin so that its quantumness is extended. People have used this sequence for more than half a century in nuclear magnetic resonance. It's known to be used to correct for driving field imperfections, so imperfections in your microwave source. But it's also known not to correct the tunings from resonance, that is the delta that you want to measure. So we can actually use the sequence to give electromagnetic kicks onto our spin without correcting for this detuning delta from the resonance that we want to measure. So if you do this, if you apply those electromagnetic kicks, this is what you recover from your quantum sensor. So here's a signal, which is basically just normalized fluorescence intensity as a function of the time during which you apply this sequence with your microwave cable. Okay? So here's what you see. For those of you who have seen this in quantum mechanics class before, this is just a Rabi experiment modulated in a weird way. If we are at the top of the signal here, with the spin emitting, the defect emitting like the maximum light, you are, say, in state 0. If you are at the bottom of the signal, you are in state 1, and every fluorescence intensity in the middle signals that the spin is in a superposition state of 0 and 1. And it turns out that we can do a theoretical method called average Hamiltonian theory to know exactly how the signal depends on this tiny little delta that we want to measure. So we know how the signal depends on delta, so that from this signal we can recover delta. And for example, we can do the Fourier transform of the signal, getting something back that looks like a spectrum. I'm not going to tell you how to read this. It's not that simple. It doesn't matter. But suffice it to say that here with this signal, we are measuring 3 different magnetic fields, the smallest one being about 1/10 of the magnetic field of the Earth. That is very tiny. Okay? So here's how we can use this spinning diamond as a very sensitive quantum enhanced magnetometer. But big picture, tiny sensor that is able to measure tiny quantities. So we come to the end of the first part of my talk. I just want to make sure, okay, that you agree with me that I've just presented you with a quantum sensor that works at room temperature and in noisy environments. In particular, one of the questions that I get, and I also get the same question for the second part of my talk, is the following. So here's what I've just told you. I've just told you that there is a 2-level system in diamond, right? That has a resonance frequency of about 3 gigahertz, and it works as a quantum object at room temperature. If you Google like KBT, thermal energies at room temperature in gigahertz units, you get the thermal energies at about 6,000 gigahertz. So here's what I just told you. There is a 3 gigahertz resonant 2-level system in a material at room temperature immersed into this very high temperature 6,000 gigahertz thermal bath. Doesn't this thing thermalize? How is that a quantum sensor at room temperature? Is there a paradox there? And the answer to that is that there is no real paradox. Yes, there is thermalization. As I told you, everything that starts quantum dies classical. But before thermalization, before the coherence kicks in, the information that I get from my object is quantum enhanced. So all the useful part, all the useful information is gotten while this spin is well described by the laws of quantum mechanics, while the spin can be remain in a superposition state. So there is no paradox there. Yes, the coherence times, as I said, for example, take about a couple of microseconds at room temperature, but after that time, we need to start again the experiment. All the quantum information, quantum enhanced information is gotten before the coherence kicks in. So no paradox there. And I hope you agree with me. Feel free to ask questions. If nobody interrupts me, I'll keep on talking. I love talking about my stuff, especially this, because...
Hello. Just a question.
There are
Quick
questions.
question.
Yes. Awesome.
Why does it take so long to thermalize?
Yeah. This material is unusually... diamond is unusually stiff, right? Also, it turns out that most sources of noise in diamond come from a nearby spin impurities in the vicinity of this defect. Most of carbon is carbon-12 without a nuclear spin, so you might need to consider the very small amount of carbon-13 that have a spin 1/2 that messes up with your spins. But so I think it's a combination of a stiffness of the material and the fact that there aren't a lot of spin impurities around. And if you will, you can think about this effect as being sort of buried in the dead at the center of the band gap of diamond, right? So it's a very unusual thing to happen, and that's why people are so interested in this defect, because it can be used, for example, to learn about, say, processes that occur at room temperature, say in biology. It's biocompatible, it works at room temperature. That's one of the reasons why this is so nice to work with this, because you don't need a vacuum chamber, you don't need a cryogenics, right? That's one of the reasons why this is a very good quantum sensor.
Okay, thank you.
So
you can continue.
Up to like about 6-7 years ago, this sensor in diamond, these types of sensors, technological sensors that you see there, some of which are quantum enhanced, these were the types of sensors that I was used to thinking about. At some point, though, I realized that nature very often produced sensors that outperformed humankind-made sensors in crazy, crazy ways. Okay? So I started looking at nature-made sensors. I'm going to spend the rest of my time here convincing you that there's a particular magnetic field sensor present in nature that works very similarly to the quantum sensor that I just explained to you. So now I'm not going to use diamonds for anything. Diamonds are not going to appear in my research, but I'm going to draw a very clear parallel between this nature-made sensor, magnetic sensor, and this technological quantum sensor that I have just explained to you about. And here's what I'm going to talk to you about. That's the part that I'm really excited about. So if we want to talk about sensors in biology, we might start with thinking about biology at the nanoscale, namely the chemistry. So it's been known unambiguously for decades that for test tube chemical reactions, there's a class of chemical reactions that are electron spin dependent. Here's how this works. Okay, so you should be looking at me now in broad strokes. Again, no doubt that this is what's happening. There's a chemical reaction that is happening, and at some point, the chemical reaction comes to a crossroads. At that point, the chemical reaction effectively looks for the electron spin state of a particular electron. If that spin is up, or in jargon, if that spin is measured by the environment to be up, the chemical reaction continues through one branch. If that particular electron spin is down, or measured by the environment to be down, the chemical reaction continues through another branch. Importantly, the macroscopic final products of those two branches are different. So it's known in spin chemistry, no doubt about that at room temperature in test tubes, the finicky spin state of a particular electron can have macroscopic consequences on the outcome of a very broad class of chemical reactions. Now, if at the point where this chemical reaction is at this crossroads, the almighty electron spin that we care about sees an external magnetic field, this electron spin will talk or sense this external magnetic field in a way that is actually indistinguishable from the quantum sensing modality that I just described to you. Chemists don't call it quantum sensing, but this electron spin is going to be altered by this external field. There's going to be a superposition state involved. And to cut a long story short, this external field will actually alter the probability of us finding the electron spin up or down. That's in this way that magnetic fields have been known to alter electron spin dependent chemical reactions for more than decades in the field of chemistry. Okay. So this again has been demonstrated at room temperature in solution in the gas phase, in the solid phase, for magnetic field strengths as small as the Earth's magnetic field, which is orders of magnitude smaller than the magnetic field that you sense when you put your cell phone close to your face. So here's how it all comes from chemistry to biology. It's all because of birds. As it's going to become clear, I don't care about birds, but birds have the big advantage of bringing this whole conversation from biology to chemistry and from chemistry to quantum. So birds, again, there's no doubt that this happens, but birds have been known to use the very tiny magnetic field of the Earth to migrate when they go north and south, at least as a partial cue. Again, magnetic field of the Earth is tiny. So how might they be doing this? In the end of the 70s, a brave, at that point, crazy theoretical biophysicist made an outrageous hypothesis. Paul Schulten hypothesized the following. Well, were this type of electron spin dependent present somewhere within the bird under physiological conditions, in the messy environment of the cell, right? If this type of electron spin dependent were to be present within the birds and other organisms in general would react to magnetic fields to the extent that they would react to the different physiological concentrations of products coming from these electron spin dependent chemical reactions. Because if the bird can sense different final products of those reactions, they can effectively sense the field. So here's a hypothesis that has not yet been proven or disproven. Well, maybe as the bird looks to the right and looks to the left, going through different magnetic field lines, well, maybe there is some sort of physiological modulation of the sensitivity to light in their eyes, so that the bird looking to one side, they see things darker, and to the other side, they see things clearer, so that they know that they should go to the brighter image or something like this. Again, hypothesis. This still, people don't have a good idea of how this happens in birds at the signal level. But experimental biophysicists liked this crazy idea at that point, and they started looking at proteins, animal proteins, because birds at that time, animal proteins that could actually sustain such electron spin dependent chemical reactions. And at that point, the only animal protein that was known to be able to sustain such electron spin dependent chemical reactions was this protein called cryptochrome. Now, cryptochrome is a fluorescent protein. It has a flavin, a chromophore, so that if you shine light at it, it gets excited and then emits light back. And a cryptochrome is present in the eyes of birds, in the antennas of migrating butterflies, and it's also present, for example, in all our human cells, all our human cells because it also has circadian rhythm regulation functions. And it turns out that cryptochrome is very well conserved throughout the tree of life. It appears everywhere from all those organisms that you see there, from algae to plants, to turtles, to insects, to humans, right? And it turns out that there, for all those organisms boxed in red, there is macroscopic evidence that those organisms respond to magnetic fields in a way that has to do with cryptochrome. So that's very good news. And indeed, over the course of more than 40 years, evidence for cryptochrome-based magnetosensing is very widespread, except at very disconnected length scales. I'm going to talk about the big scale experiments in a second, but I want to draw your attention to experiments made with cryptochrome in solution, the evidence of which you find to the left of my slides. For cryptochrome in solution, there is absolutely no doubt that you have this protein that is acting as a bona fide quantum sensor at room temperature. For example, researchers took a vial of cryptochrome, shone laser to excite the chromophore in the protein, and captured the fluorescence here in the top left plot, captured the fluorescence coming out of this vial of cryptochrome as a function of the time during which you shone the laser at it. The first thing that you see is that this fluorescence is going down. You're killing the chromophore. You're effectively decreasing you're damaging your proteins, right? But at the same time that you're damaging the stuff, before your damage is complete, if you pulse, and that I don't show, if you pulse a magnetic field, tiny magnetic field on and off as you're capturing this fluorescence is responding by changing its mean intensity in a way that follows the field. This is not a blip. This has been repeated with different magnetic field strengths and frequencies. And what you see is a steady state change in fluorescence intensity as a function of magnetic field strength. Now, this is in analogy to the spinning diamond that I explained to you in the beginning of my talk. Just by looking at how strongly it was emitting light, you could do quantum state readout. You could actually infer if the spin was up or down. Here, just by looking at how strongly the vial of protein is emitting light, you can actually infer if the electron spin of interest was up and the reaction continued to this branch, or if the electron spin of interest was down and the chemical reaction continued to this other branch. So that's really bona fide quantum sensing. And there is also evidence. The evidence is not extremely good, but it's not that bad either. But there's evidence that electrons in cryptochrome remain coherent at room temperature in test tube in solution for about 1 microsecond, which is also extremely long. In cryptochrome, it's not super clear why that's the case, what makes a cryptochrome unusually special in terms of the coherence. But just to give you an idea of how long that is. So there are much better ways of quantifying this, but back of the envelope calculation is the following. Electrons, when they are in a magnetic field, they spin precess, they Larmor precess. So very back of the envelope calculation. This is like a super bad bound, but you can say that an electron can sense a magnetic field if it can Larmor precess once around the field before it decoheres. This is an overkill. I mean, things are much better. We can do much better, but order of magnitude. For a free electron precessing under the Larmor frequency of the magnetic field of the Earth, okay, which is about 1.4 megahertz, you get a period of about 750 nanoseconds. This means that for an electron in cryptochrome to be able to sense the magnetic field of the Earth, it should remain coherent for about 715 nanoseconds. And now there's evidence that it can, in principle, stay coherent for 1 microsecond. So this protein looks good for being able to sense the tiny magnetic field of the Earth. So that's all consistent with the quantum sensing picture. Except that the next available level of evidence comes at very, very large length scales in beautiful but hard experiments performed by biophysicists, biologists, and chemists. So, for example, it's people who catch 20 birds during their migration, put them into cages, put them into magnetic fields, mess up with the magnetic fields that the birds see, and they see that when they change the magnetic field, the birds want to go out of the cage via different directions in a way consistent with this electron spin dependent chemical reaction. Flies are not known to migrate, but they can be trained. I have no idea how biologists do this. This is harder than quantum mechanics for me, but flies can be trained to find food in the presence of a magnetic field, right? So they train the flies to do this. Then the researchers knock out or remove the cryptochrome genes from the flies, and the flies are no longer able to find food. They're no longer able to be trained to find food when a magnetic field is on. In further experiments, researchers put back human cryptochrome inside the flies, and the flies are back to being able to be trained to find food in the presence of a magnetic field. And there's many more examples that I'm going to talk about, but all of this macroscopic data is consistent with this quantum sensing picture that is unambiguous at the test tube level. But to this date, on Earth, there is no experiment that can unambiguously prove or refute that a single superposition happens or let alone is being used inside a single cell. Can we do better? Can we unambiguously link those two length scales? And I am here to say that that's what we are trying to do. So we want to bridge those length scales by building what I call glorified microscopes with coils. So what we want is to bring techniques from the quantum sensing realm to bring the same type of instrument rigor that we have, that we would find in a quantum optics, quantum sensing, quantum computing experiment, right? But make those experiments work for biological samples. What we are starting to do with our big experiments is trying to apply the same quantum control techniques. Instead of controlling a spinning diamond, controlling a spin inside a protein, say inside a single cell. Again, the readout of the spins is done via the same way that one would do for the spinning diamond, is based on fluorescence intensity differences, is the quantum state readout, and we want to see how a different initialization, manipulation, and readout and of these particular electron spins, how they affect downstream cellular processes in the same experiment using regular fluorescence techniques that people have been using in the field of biology for a long time. It doesn't really matter. I'll tell you more towards the end. So our experiments, they don't look like regular microscopes where you look at, they look like big optical tables with plenty of electronics. The instruments are being set up to be synchronized down to the nanosecond. And I think that the experiments that exist right now for this flavor of quantum biology, they're necessary, but they're not sufficient. The experiments that are going to be sufficient are the ones that can unambiguously prove or refute that superpositions, spin superpositions are actually happening, can actually be sustained in cells for long enough for them to be able to sense tiny magnetic fields. The other cool thing is that differently from many biophysics approaches where you observe a phenomena and then model, here, using the tools from quantum mechanics, for example, the tools of open quantum systems, you can do sort of crappy simulations that can start to give you an idea of what you can expect from your experiment. So the mathematical models can actually drive your experiments. So I won't have time to describe in detail, maybe during the question part. But for reasons that are well understood in the spin chemistry of these electron spin dependent chemical reactions, the magnetic fields that actually change the course of these chemical reactions are relatively small. So there's an interplay between hyperfine interactions and external magnetic fields. I'm not explaining. You ask me in the questions. It's a subtle phenomenon, but the magnetic fields that can actually actuate on this class of chemical reactions are relatively small fields, on the order of the Earth's magnetic field, on the order of the magnetic field produced by your cell phone. This also means that if you put the magnetosensitive vial of proteins inside a 3 Tesla magnetic resonance imager in the hospital, the 3 Tesla is not going to be able to alter the fate of the chemical reaction. So magnetic fields that matter are tiny. And this is well understood, but you can also see this in the simulation. So for example, have a look at this top right plot here that our group did, but other people did as well. This is the simulated, I'm dying to take this curve experimentally, but this is the simulated magnetosensitivity of a vial of cryptochrome for cryptochrome tumbling in solution. So here's how much it responds to a tiny magnetic field change as a function of magnetic field strength. And as I mentioned, the dependence on the field strength is not monotonic. It goes up and down with field strength at about 10 times the magnetic field of the Earth. At about 600 microtesla, the magnetosensitivity is already very much reduced. And what's cool is that, again, not a biologist here, but this magnetosensitivity simulated peaks very close to the magnetic field of the Earth, which might be a coincidence, or it might be that there might have been some evolutionary pressure for those proteins. If they're really helping birds and other organisms navigate using the magnetic field of the bird, there might have been evolutionary pressure for those proteins to evolve to function very close to where they should function. We're just physicists. We cannot tell the difference if it's a coincidence or not, but we think that people who do directed evolution of proteins can help us say if proteins can react to this sort of evolutionary pressures for magnetic fields. So that's very interesting. But in the last 10 minutes of my talk, I want to tell you that it's not only about birds. That's the most important part here. There are many, many other types of biological responses to weak magnetic fields that are consistent with being caused by this type of spin-dependent chemical reactions, influencing all types of relevant physiological phenomena, from the production of cellular oxidants to yield of DNA repair to up and down regulation of cellular proliferation rates to cellular motility to changing the fate of stem cells, all in a way that is consistent with those effects being caused by this type of spin-dependent chemical reaction. I can cite a lot of examples, but one picture speaks more than words. So have a look at this bottom picture here that I have authorization to show. So I have a friend. His name is Peter Schierlinger, and he is a bona fide precision measurement physicist. He's in Munich, and what he does for a living is the following. He builds very good Faraday cages, usually referred to as hypomagnetic chambers, that can shield very well external magnetic fields. Usually he shoves ultra-cold atoms experiments inside, and they can be very well shielded from the magnetic field of the Earth. He's like world expert in this. So magnetic field of the Earth is about 50 microtesla. Inside his chambers, he can have noise levels DC of about 1 nanotesla. So he started hearing things about, electromagnetic field effects in biology. So here's what he set up to do. For 2 days, he grew tadpoles or frog embryos, girinus, inside those hypomagnetic field chambers under 2 conditions. Now this has been reproduced by 2 other groups. This seems to be a thing. In one group, the tadpoles were grown with a field applied inside, mimicking the magnetic field of the Earth, which is super tiny, right? Just with a simple coil. After 2 days, the tadpoles were okay. However, if a new batch of tadpoles were grown just under the nanotesla residual field, the result is what you see macroscopically there. About 35% of the tadpoles are actually malformed. Okay, the embryos are no longer viable. So have a look at what I just told you. I just told you that you don't even actually need to apply a magnetic field. You can take out a magnetic field and you mess up with embryology. Again, this is not the first example of that. People have been seeing those correlative results for 40 years. Okay, data piles up, and because in this case the field intensities involved were so tiny, right? Everyone's best guess is that this is not, if this could be explained by a big chunk of magnetite or a tiny magnet inside the tadpoles, those chunks of magnetites to respond to a difference in field, this mole would have to be huge, and those huge chunks of magnetite are not known to happen in tadpoles. So everyone's best guess is that this must be a spin-dependent chemical reaction. So my best guess is that this has to do with melanin. Melanin is the thing that gives our skin its color, and it gives the tadpoles skin like their color. And melanin, there's very rich spin physics in melanin. So again, it's not only cryptochrome, it's not only melanin, there's a broad class of proteins that can sustain such electron spin dependent chemical reactions. And again, this opens up a lot of crazy questions, like a Pandora box of questions ranging from, say, space exploration, right? So what's the magnetic field on Mars? Can we reproduce on Mars? Can you space farm on Mars? Mars has a very weak magnetic field compared to the Earth. So I've been trying to tell NASA people that it's not only microgravity, it's not only cosmic rays, you need to start thinking about magnetic fields. Two other things like therapeutics. Can you learn how to deterministically change this endogenous, this native quantum knob inside cells using weak magnetic fields for, say, therapeutic applications? Can you deterministically learn? It's a new way of actuating on biology. A very recent result has shown that ion channel functioning. So, for example, all our processes in the brain, they're regulated by ions going in and out of cells. Ion channel functioning can be regulated by weak magnetic fields in a way consistent with this type of electron spin dependent chemical reaction driving the process. So, can you control brain processes with weak magnetic fields, for instance? That's one of the type of things that we think about. So let me tell you where we're at. We're not there yet. The timeline of my lab has been from hell. Okay, so we're a little bit closer to the middle now of 2023, but it has been a mess. Nothing in my life has been normal since I started at UCLA. Importantly, we're still building our big quantum experiments, and we've had a lot of issues regarding orders. So while we're building our big experiments, we are still taking better, more correlative data of magnetic field effects in cells. And again, we've only been approved for cell work since January, so this is very recent and first results for us. We have built very cool water-cooled coils that are super controlled, automated, and we can put those coils inside an incubator and actually grow cells with very controlled magnetic fields for different durations, different strengths in a physics-approved way. So we really like those coils. Those coils are an improvement over existing coils used by biolabs to perform weak magnetic field experiments. And with those types of coils, we have already gotten correlative data in our own quantum experimentalist run lab. For example, let me tell you what, for example, magnetic fields on the order of 6 times that of produced by your cell phone can do to, in this case, rat muscle cells. For example, after 4 hours of exposure, we can see things such as cytoskeleton, cellular cytoskeleton changing because of this magnetic field, the way that the cellular nucleus change as a function of magnetic fields in a reproducible way. Cytoskeleton in cells is composed of two types of polymers, actin and microtubules. Microtubules polymerization rates, like how much they aggregate, also seems to change as a function of magnetic field. And again, last thing I want to show, this is one data set, it's not yet reproduced. It's the same data. To the right is the way the chemists plot them, to the left is the way a physicist would like to see things plotted, but the idea is the same, that very weak magnetic fields also seem to be altering. I'm not telling you how we do this in the lab, how we measure these rates, because it's a bio process, ask me later, but rates of production of this H2O2, the cellular oxidant, can be changed as a function of magnetic fields in a way that would be expected by this type of spin-dependent chemical reaction. So the problem is that right now, unless we build those big quantum-like experiments that can provide, if you will, an idea of the local spin Hamiltonian that this spin inside a protein is subject to, we're looking for a haystack in the dark. We chose those magnetic field parameters because of reported literature, right? But right now, there have been people looking at those magnetic field effects in a heuristic way for 40 years. We need to go past that. We need to start doing things deterministically. And we think we can do that with those very controlled quantum-like experiments that we're still building. But in the end, the final product where we want to be, in the end, we won't need those big quantum experiments. The final product is already achievable with like your cell phone, right? So where I want to be, completely science fiction at this time, is like I have an app in my phone, and then I say, well, today I need help with wound healing. You go to your app and you press a button for wound healing, and your phone produces the exact magnetic field strength and frequency that you need to do wound healing, and then you do like this. It's another way of actuating on biology, and we just need to understand this knob that is there, even if it's not quantum. This knob is there indisputably, and we need to understand how to control it deterministically. I'm not going to have to cut a long story short, we're building experiments, the big quantum experiments. We don't have results yet. They're slowly being built, but I would like to thank the wonderful people in my lab who have been making this new science come true. So I end in the way that I always end my talk. So may the quantum be with you. What are your questions?
So thank you, Clarice. So finally, the so-called myth that it might be bad to sleep next to your cell phone might not be a myth after all, right?
So here's my take. Yeah. Each chemical reaction, I don't think people should panic. Like each chemical reaction is driven by a very specific magnetic field intensity and frequency, right? So the chances that your cell phone manufacturer was either lucky enough or unlucky enough to use that exact same magnetic field intensity and frequency, I think those chances are small. So, and I think that at this point, the chances that your magnetic field are doing your cells good are the same that they're doing your cells bad. They might, because those processes, they can up and down regulate it. That's the beauty of that, right? In the sense that so the military are interested in this because it might enhance the performance of soldiers, right? The people in therapies are interested in that because it can downregulate how fast cells divide in cancer, right? So those things can be modulated up and down, and that's the beauty of it, I think.
Okay, so do you have, I'm going to pass by to questions in the auditorium. Do you have questions? So one here.
Hi, thanks for this nice talk. I don't know if you can switch to Portuguese. Now.
Yeah. Oh, sim, vamos.
Tá, você tinha mostrado alguns exemplos dos girinos que, na ausência de campo magnético, eram prejudicados.
Isso.
O campo magnético da Terra ele já inverteu várias vezes ao longo dos milênios. há algum registro de como isso afetou a vida na Terra?
Essa é uma questão cuja resposta eu não sei. Muitas pessoas já me perguntaram, eu já fui atrás disso, e em resumo, eu não sei. Aqui é o que se sabe. Normalmente, quando o campo magnético da Terra, não sei se inverte ou muda, isso está correlacionado com mass extinction events. Mas também pessoas do campo de geofísica já me falaram que tem outras possíveis explicações para esses mass extinction events. Ou seja, sim, quando o campo magnético da Terra muda, normalmente aparece junto mass extinction, mas a gente não sabe se é por causa desse tipo de fenômeno ou não. Mas é uma questão muito interessante, eu acho que pessoas tinham que ver. E outra coisa interessante, por exemplo, é assim, o campo magnético da Terra está sempre mudando, né? Será que dá para a gente fazer um registro de como as rotas migratórias sei lá, das borboletas, dos passarinhos muda, né? Quando esse campo da Terra vai mudando? Eu acho que isso seria um projeto de longo termo bem interessante também.
Temos mais perguntas no auditório?
Clarice, muito obrigado pela palestra. Eu queria saber se vocês fizeram algum experimento em que você tem as medidas controladas do seu campo magnético muito pequeno e você coloca células vivas e depois células mortas, para ver se há diferença.
Não. Tá aí um bom controle que a gente podia fazer.
Eu acho que para ver se as células afetam a sua medida. E se tem diferença entre as células mortas e vivas.
Boa. Por exemplo, tem coisa que eu acho que a gente não consegue fazer. Eu acho que, por exemplo, esse experimento depende, por razões que eu não expliquei, depende das células estarem vivas e produzindo coisa. Bom controle. Nossa, obrigada. Gostei da ideia porque eu acho que é um negócio que eu nunca vi na literatura. Muito boa ideia. Muito legal. Como que é seu nome? Eu não consigo ver quem que está falando.
Eu não sei, olhando para cá, talvez você veja?
Não, para frente é melhor. Isso, eu estou te vendo. Tanto faz.
Eu sou Oswaldo, professor aqui do Instituto.
Obrigada, Oswaldo. E o outro professor que fez a pergunta antes, que tá lá. Oi, como que é seu nome?
O Neto, professor Manaus.
Manaus, ok.
Vários nomes.
Obrigada. Ok. Eu acho que tinha uma outra pergunta.
Bom, eu já começo dizendo meu nome, então.
Isso.
Walter. Obrigado, Clarice, pela apresentação. Boa, muito interessante. Talvez eu tenha perdido alguma pequena parte na sua apresentação inicial, mas a minha pergunta ou minha preocupação é a seguinte. Você tem um quantum sensor lá que está estudando o seu sistema de 2 níveis. E eu suponho que do quantum sensor até a sua bancada deve ter um outro registrador, um outro sistema macroscópico que recebe o sinal de lá, do sensor, e é desse sinal que você vai fazer a sua análise. Estou certo nesse raciocínio?
Sim, certo. Sim. Por exemplo, aqui. Por exemplo, o nosso sensor, no caso do diamante e no caso dos experimentos que a gente vai fazer, tá gearing up para fazer, o nosso sensor vai ser um sensor de single photon.
Certo. E a segunda parte da minha pergunta é a seguinte. Uma vez que você tem, então, um quantum sensor, eu suponho que você deva ficar preocupado com o nível de emaranhamento dele com o seu sistema, porque o seu registrador, na verdade, não deve estar registrando a informação do seu sistema, mas do conjunto sensor e sistema agora. Então, eu acho que o emaranhamento dos dois é que, na verdade, você deve estar registrando no final. Entende? Porque o sensor agora me parece tão quântico quanto o seu próprio sistema. Então, os dois formam um sistema só.
Eu concordo. Eu não sei se eu não tenho um argumento. Eu concordo absolutamente com você, mas assim, nossa, eu não sei como. Eu concordo né? Mas assim, pensa em experimento, isso não é diferente de experimento com átomos frios, por exemplo, né? Que a gente pode também fazer experimento e depende da intensidade de fluorescência. Por que você acha que é diferente esse experimento com esse sensor quântico de um experimento, sei lá, num vácuo com uma cold cloud,
Bom,
por exemplo?
Não sei, também não saberia dizer, mas quando você coloca esse adjetivo quantum no próprio sensor, me parece que seria meio que difícil de distingui-lo do próprio sistema que você está estudando devido à própria natureza minúscula que ele deve ter em contato com o seu sistema.
Um outro jeito
ele não é mais uma
de
espécie de banho da coisa, ele é o próprio, ele faz parte. Como se um átomo estivesse como sensor de um outro átomo. Na verdade, os dois estariam emaranhados.
Eu acho que sim, e eu acho que assim, no momento que a gente recebe a informação nesse single photon detector, o sensor do diamante está projetado.
Certo.
Então talvez isso resolva um pouco, que no momento que a gente vê a fluorescência não tem mais... Bom, não sei. Não sei se eu consigo responder.
Tudo bem, obrigado.
Temos mais perguntas? Eu tenho uma pergunta, Clarice.
Sim,
você
mande.
mencionou que uma das... posso fazer? Você mencionou que um dos aspectos que permitiam você fazer medida, né, de quantum sensor em diamante era uma característica muito específica do diamante dele demorar para se termalizar. Como é que você justifica, como é que você pode... Esse mesmo efeito existe também de um tempo de termalização muito longo, também existe no caso desses objetos biológicos, dessas soluções biológicas?
Então, para o criptocromo, para o que está vendo o campo magnético da Terra, de novo, tem dado que indica que o tempo de termalização, o decoherence time, né, do elétron no test tube é aproximadamente um microsegundo, que é enorme, por razões que não se entendem. Olha como eu vejo essa coisa. O campo magnético da Terra é muito pequeno, né? Você só precisa ter tempo de decoerência longo se você quer fazer uma medida muito pequena. Talvez os campos magnéticos a que as outras proteínas respondam sejam maiores, então você não precisa ter esse tempo de decoerência tão longo, né? Você pode ter um sensor que é mais ruinzinho, né? Que só sobreviva por agora eu estou especulando, completamente especulando. Eu acho que, por exemplo, cada tipo de proteína pode, do mesmo jeito que talvez o criptocromo seja optimizado para funcionar no campo magnético da Terra, outras proteínas sejam optimizadas para funcionar sentindo, né, fazendo o sensing de outros campos magnéticos que talvez sejam maiores. Né, por exemplo, de novo, especulação total. Os nossos processos celulares são regulados pelo fluxo de íons entrando e saindo da célula. Talvez esse fluxo de íons, que é muito bem regulado em duração e intensidade, né? Talvez esse fluxo de íons estejam produzindo campos magnéticos que influenciem algum tipo de electron spin dependent chemical reaction na célula, né? E de novo, talvez a gente se sabe que cada reação química funciona bem, funciona de maneira optimal para uma combinação particular de intensidade e frequência, né? Então, talvez já haja essa autorregulação que ocorra na natureza, mas mesmo se não houver, mesmo se a natureza não estiver autorregulando essas coisas, ou mesmo se a natureza não estiver usando isso, o meu argumento é que a gente pode usar isso para modificar a biologia, né? E, de novo, mesmo que não seja quantum, mesmo que não seja o spin, que não tenha a ver com o spin, evidência que campo magnético fraco está alterando biologia de maneira drástica existe existe por mais de meio século, né? No Ocidente, né, electromagnetics in biology é meio tabu, né? Pesquisa em electromagnetics in biology acho que foi meio contaminada por, sei lá, New Age, não sei o quê, né? Então, mas no Oriente, na Rússia, no Japão e na China, essa pesquisa nunca parou, né? Então, por exemplo, o Japão é uma das maiores potências de spin physics. Tem muita gente fazendo spin physics no Japão, né? Que é exatamente isso, electromagnetics in biology, né? E eu acho que biologia hoje é muito fixada nas leis de Newton. Eu acho que, pelo menos, mesmo se não seja quantum, a gente tem que fazer o shift para também incluir as leis de Maxwell, né? Em biologia, em algum jeito de atuar em biologia usando um outro tipo de botão.
Obrigado. Só uma perguntinha que alguém me perguntou. Você tem ideia, você mencionou o fluxo de íons, né, nas células. Você tem ideia da intensidade do campo magnético que isso gera?
Tenho, porque, aliás, no meu paper do diamante, no meu paper do diamante, eu fiz a comparação e, assim, eu sei que o sensor de diamante consegue medir o campo magnético da ordem desses produzidos pelo íon. Como o sensor do diamante parece ser mais ou menos da mesma sensitividade do criptocromo, por exemplo, eu acho que o criptocromo poderia sentir esse fluxo de íons. Eu não sei outras proteínas, mas o fluxo de íons, eu posso mandar, se alguém me mandar um e-mail, eu posso mandar, eu fiz essa comparação em algum momento da minha vida.
Tá bom. Tem mais perguntas, Cibele?
Oi, Clarice, obrigada. Meu nome é Cibele.
Oi, Cibele.
Se eu entendi direito, você falou sobre o criptocromo e ele está em todas as células do corpo humano?
Sim.
E aí, no caso da borboleta, também está em todas as células ou ele fica localizado em alguns lugares específicos?
Então, da borboleta eu não sei. Eu sei que a parte da... Então, para a borboleta eu não sei. Borboleta, parece que a sensitividade do campo magnético vem da antena. E se sabe que tem criptocromo na antena. Para o pássaro, eu poderia especular que o pássaro também tem criptocromo em todas as células, né? Porque a gente está perto, né? Na cadeia biológica. E assim, o negócio é... No pássaro, o criptocromo que parece que está envolvido de sentir o campo magnético na Terra é o criptocromo do olho do pássaro, né? Uma coisa que eu não mencionei é que muitas dessas reações que são electron spin dependent, elas são reações que começam com a absorção de um fóton pela flavina, pelo pigmento, pelo cromoforo da proteína, né? E o lugar onde você tem o maior fluxo de fótons é no olho, então teria uma vantagem dessa reação para sensitividade do campo magnético, dessa reação está acontecendo no olho. Outra coisa que eu queria mencionar é que daí você pode falar, então se todas as reações são fotorreações, qual que é o interesse das nossas células de dentro do nosso corpo terem essas proteínas? Né? Uma coisa que não é muito mencionada, e de novo, não tem nada de esquisito nisso, é que o byproduct de muita reação química é fóton, né? Então, isso não sou só eu que faço essa hipótese, tem muita gente que faz essa mesma hipótese, que talvez esses byproducts dessas reações que ocorrem, químicas que ocorrem em todo lugar no nosso corpo, em biologia, talvez esses fótons estejam sendo absorvidos, sei lá, pelo criptocromo ou por outras proteínas, para começar esse ciclo de sensitividade do magnetic field, mesmo para proteínas que não são exteriores. Não é a melanina, não é o criptocromo no olho, né? Então, eu acho que tem um monte de coisa de biologia que a gente não entende. Por exemplo, uma coisa que eu pergunto para muito biologista é que cromoforo, né? Tem muito cromoforo, de novo, em célula do interior. Tem cromoforo que é achado em alga do fundo do Mar Vermelho, que nunca viu luz na vida. Por que a evolução por que não caiu fora da evolução esses cromoforos? Por que cromoforo continua sendo achado em organismo que não vê luz, que não precisa de luz, né? Assim, não precisa de luz. Então, e as pessoas não sabem, a gente não sabe se ainda não caiu da evolução ou se é um negócio que está sendo usado de uma maneira que a gente ainda não entendeu.
Muito obrigada. A minha pergunta era, eu ia fazer uma pergunta bem mais singela, mas agora fica até, ia fazer uma pergunta meio boba, até comparada com tudo que você falou.
Manda.
Não, achei
Não
muito
existe
interessante.
pergunta boba.
Eu ia perguntar assim, se eu não entendi errado também, você falou que eles fizeram testes nas borboletas, e aí eu entendi que era tirando o criptocromo.
Na flies, em mosca, mosquinha.
Tira o criptocromo, então, porque daí então nas moscas se sabe exatamente onde eles estão localizados, é isso?
Eu não sei, eu não entendo nada de biologia, eu sou física. Eu não sei se eles conseguem tirar o criptocromo só de alguns lugares particulares, mas eu sei que em mosca tem criptocromo no sistema nervoso. Que tem a ver com circadian rhythm regulation. Em resumo, eu não sei se no caso das moscas eles tiram de todas as células ou eles tiram de só algumas células. Eu não sei direito
como... Mas está ótimo, obrigada. E deixa o resto do pessoal perguntar também. Obrigada.
Eu não estou ouvindo nada, gente. Não sei se é...
Bom dia, professora Clarice.
Bom dia.
Parabéns pela apresentação. Eu entendo que você é física.
Não, você não falou seu nome.
Meu nome é Altamir. Sou aluno daqui do Instituto. Bem, é interessante quanto à circulação de íons, né, que lembra espécies reativas, radicais livres, de certa forma. Né?
E isso tudo é radicais livres. Isso tudo que eu apresentei é radicais livres.
Sim. Aí a questão, sabido que o processo de respiração celular ele pode ser afetado pela capacidade ou função de reparo do ADN, DNA, né? Então, assim, há alguns desenvolvimentos assim sobre a questão da função de reparo e como campos magnéticos afetam.
Sim. Por exemplo, tem um paper que mostra que respiração celular é impactada em placas de célula, que a respiração celular é impactada por campo magnético fraco. Outro, isso aqui é em bactéria, mas DNA repair em bactéria é alterado por campo magnético fraco. Tem um monte de dado sobre isso. Correlativo, né? Tudo isso é correlativo, mas sim, tem muita correlação com o que você está falando. Já tem gente vendo isso, e eu acho que essa é uma das outras coisas que vai explodir em breve. É gente tentando fazer regulação de radicais livres com campo magnético. Sim.
Temos, acho que mais tem uma pergunta do auditório, mas temos uma pergunta aqui antes do professor Sérgio Muniz no YouTube. Ele passou uma excelente palestra, Clarice, um prazer vê-la novamente, aparentemente te conhece. Uma dúvida, qual é a frequência de ressonância da proteína criptocromo e qual a região espectral de absorção e fluorescência?
Tá, então, primeiro o negócio de frequência. O modelo né, que a gente faz com o spin prevê dependência em frequência, em intensidade do campo e para a proteína que está em membrana de direção. Eu vou falar da frequência. Frequências de interesse podem ser desde DC field, né, só um delta no field, até frequências que que tem alguma coisa a ver com a spin physics. Por exemplo, kilohertz que tem a ver com ressonância de spin atômico, megahertz, que tem a ver com Larmor frequency, né, de elétrons. Então, por exemplo, a simulação que eu mostrei aqui, né, é para um DC field, mas tem outras simulações que dá para fazer como função da frequência desse campo magnético, né? Hoje em dia é tudo heurístico, né? Você tem a simulação e você tem um monte de ponto, né, super esparso nesse phase space, que é intensidade, direção, frequência, e não tem muito modelo determinístico para entender o que está acontecendo, né? Por que a célula responde tanto com o DC field nessa intensidade, como com 300 kHz nessa outra intensidade, né? O que se acha é que essas frequências de interesse, quando não são DC, tem a ver com a spin physics, tem a ver com Larmor frequency de electron e nuclear spins.
Obrigado. E só uma outra pergunta dele também, se há diferenças significativas na fluorescência entre experimentos feitos com criptocromo puro em solução com relação à proteína em células.
Esse que é o problema. Em células não existe esse tipo de dado. Né? Esse que é o negócio. Né? É isso que a gente está tentando fazer, porque esse tipo de dado em célula mostraria que uma superposição pode tá existindo em célula. Hoje em dia, de novo, não existe nenhum experimento no mundo que mostre que superposição esteja sendo, que superposição sobreviva às temperaturas celulares, ao environment celular. Ponto. Né? Todo dado que se tem é correlativo. E é por isso que se precisa de dados exatamente como esses, mas em células. Né? Eu vou falar uma coisa sobre o nível de fluorescência em geral, né? Que é válido também para o diamante. Por exemplo, a diferença da fluorescência do diamante no nível 0 para o nível 1 é menos de 5%. Né? Então, assim, a diferença de fluorescência é muito pequena, mas se você tiver experimento suficiente, né, o diamante suficiente para rodar o experimento muitas vezes, ou se você tiver muitas proteínas para fazer o photocycle, você pode conseguir consequências macroscópicas. Para o criptocromo também é esse mesmo nível de diferença de fluorescência. Fluorescência nos diferentes spins é menos de 5%. Então é pequeno, né? Mas é mensurável e se assume que seja suficiente para fazer a diferença nessas reações químicas.
Tá, ok, só uma última pergunta aqui para...
eu tenho uma pergunta, então eu só queria entender antes uma coisinha.
Oi, qual que é o seu nome? Você não falou o seu nome. Matheus, fala, Matheus.
É assim, pelo que eu entendi sobre a parte de sensoriamento, quando a gente citar o tempo de coerência, fala sobre o tempo de coerência do sensor, né? Então, o sensor, para ele conseguir realmente fazer a medida, o sensor quântico, ele precisa ter um tempo de coerência longo o suficiente, né, para poder interagir com o campo magnético e tal, e aí conseguir ter essa relação. E aí, nisso, então, vem minha pergunta. A gente vê que animais e tal, eles têm essa interação de campo magnético, por exemplo, quer dizer, então, e se, por exemplo, um pássaro, alguma borboleta migra, ele precisa ficar fazendo essa medida constante, né, para saber sempre para que direção que ele voa. Então, assim, no caso, assim, se a gente conseguisse provar que realmente é possível manter propriedades quânticas, coerência, emaranhamento em seres biológicos, quer dizer, então, que...
Eu tenho que fazer um parênteses. Eu estou apostando minha carreira que superposição exista. Eu não estou convencida que emaranhamento sobreviva em ambiente celular. Eu acho que não existe dado suficiente para... Fechando parênteses. E para fazer quantum sensing, você só precisa de superposição. Você não precisa de emaranhamento em princípio. Então, eu aposto em superposição, eu não estou pronta para apostar em emaranhamento. Fecha parênteses. Continua, desculpa.
Perfeito, obrigado. Mas, então, assim, se a gente conseguisse dados que realmente superposição sobreviva nesse tipo de sistema, Então, quer dizer assim, por exemplo, como os animais estão sempre fazendo essas medidas, quer dizer então que existe uma chance, por exemplo, de células de animais e tal sempre ficarem produzindo estados quânticos para sempre ficar fazendo essas medidas, sabe, como se fosse uma produção constante de estados quânticos?
Isso, porque, assim, assim, o mecanismo é o mesmo com o diamante. A fotofísica é muito parecida. Eu não expliquei isso, não tive tempo, mas tudo começa com a absorção de um fóton, né? E daí, dessa absorção de um fóton, você entra nesse ciclo de transição entre um spin e outro. E a ideia é que se você está no spin, você demora um tempo X para voltar para o seu ground state. Se você está no outro spin, você demora um tempo Y para voltar para o seu ground state, né? E assim, a ideia é que quando você volta para o ground state, você está pronto para recomeçar o seu ciclo, para reentrar nesse sensing process.
Tá
Né?
ótimo.
Então, é um negócio que se auto-recicla.
Tá ótimo. Então, Clarice, acho que não temos mais pergunta. Queria agradecer mais uma vez a Clarice por ter aceito o nosso convite. Agradecer um pouco a Clarice.
Obrigada, gente. Se vocês tiverem perguntas, por favor, mandem e-mails. Foi um prazer falar com vocês.
Tá, ok. Por algum motivo, acho que sua câmera caiu. Não sei se foi.
Não, bom, tá bom.
Tá
Eu tô aqui, gente.
Tá aí.
Até a próxima, então.
Tchau, Clarice.
Obrigada,
Muito obrigado,
gente.
tá?
Tchau, tchau. Até. Boa sexta-feira. Bom fim de
Você
semana.
também.
Tchau.